Marcel. And Lucas. Okay. We have the main speakers. So I'm gonna start and then I'll hand it to you, Lucas and Marcel. Okay, so our agenda for today was slightly modified. Oh, first of all, we plan to record this meeting for some of you that are joining us for the first time and publish this on YouTube. So if anything occurred during the meeting, please let us know and we can make edits before. But please reach out at the end if you want something else to edit out. Okay. So our meeting agenda for today is a follow-up on the receptor field characterization in the pilot that Lucas and Marcel led the analysis. This is a very important meeting because we are actually ready to go for collecting the bulk of the full dataset, the 2-photon pipeline and the electrophysiology pipelines. Some of the mice are ready to come to us, and so we probably are going to record in a week or two some of the first experiments. So after this meeting, I'm hoping to push out the latest modification, if we need any, of our stimulus, so that we can collect the full data set. So this discussion is important. As a result, in agreement with John Meng and Lucas, we decided to extend this meeting to the full hour. And then next week, John Meg is going to present his novel publications on mismatch responses. So today is going to be a focus on that. So maybe you want to take it from there, Lucas and Marcel? I'm not sure in which order. Oh, we can't hear you. Am I the only one? Oh, I can hear Lucas.
Can't hear him either.
Oh, okay.
That work?
Oh yeah, better.
Okay, perfect. Yeah, sorry. This now works. Yeah.
Yes, it works.
Perfect. All right. Good. Can you see the presentation?
Great, perfect.
All right. Okay. So this is mainly Marcel's analysis. I have to say, I'm just giving kind of the intro, what we are trying to achieve here, and then he will take over for later parts. Sorry, last it was shifted 1 week, so I couldn't arrange.
Incredibly thankful that you accommodate the West Coast time, Lucas.
So the kind of the default that we have. So we have the stimulus block that has the aim to map receptor fields and uses local gratings. And these gratings are shown at a grid. So these are different grid positions on the screen. This is just to show the approach. It looks a bit different in the actual data. This is just to explain how it works. And then this, for example, allows to find significant responses by counting essentially spikes when the stimulus is shown at these different locations. One can smoothen this or threshold, and then also look at different clusters here to say there's a receptor field with a certain position and size. So this is the kind of information that we can pull out with this analysis to see if visual responses occur where we would expect them. As essentially categorize the units in the recording. This is within stimulus tailored for this, but there's this new interesting approach by Sophie and Max, who are also here in the audience, and they developed this approach to map multiple properties of visual responses with one stimulus that's essentially shown here, and it contains different spatial and temporal frequencies, sharp contrast, and thereby could potentially also yield the same information as the more localized stimuli. But in order to extract that information, one uses an analysis that uses so-called wavelets. So you essentially filter that stimulus with such a Gabor wavelet that has a certain position and orientation, plus different size and frequency. You filter that movie, essentially, and then you get amplitudes per frame. And then you can ask, how is the response of a neuron correlated with the amplitude of a particular filter? So if you want, you can think of it as filtering that stimulus with the Gabor at this particular position, and then you might hope to get something like, if you look on the local scale, like a stimulus, like the localized grating, and by filtering it and then correlating with the neural response, you might actually see that a neuron, for example, correlates with a particular filter at a particular position and orientation similar to the grating stimulus. So the kind of goal of this endeavor was to compare a map of correlation with this amplitude to essentially this response raster we've seen with the local gratings. And just to see if the responses we get to the local gratings agree with what we expect, Marcel computed these average Z-score spike responses to the local gratings. And on the X and Y axis, we have the horizontal and the azimuth position. And as you can see, this is essentially spike counts here, the color maps. So we have most of the responses averaged over all units at the center of the screen, which is kind of what you would expect. This is both for probe C, which is V1, and probe D, which is LM, area LM, as I recall. Correct me if I'm wrong. And this is, yeah, we essentially see the same thing for both probes. This is good, which means that essentially at least the alignment and so on might seems to work with the local gratings. Now what we did is we selected a set of units that we found had significant responses to the local gratings. That's shown on the left-hand side for each of the columns. And then we did this for 0 degrees gratings, for 45 degrees gratings, and 90 degrees gratings, and compared that to the correlation matrix that we obtained with the wavelet transforms. In this entire wavelet analysis was kind of adapted to match roughly the same spatial resolution that we had with the gratings, as well as the same orientation. So we only used wavelets with these 3 different orientations. The size of the wavelets was optimized, but the spatial frequency was also taken to match what was shown with the local gratings. However, what we didn't really see a correspondence here between the receptive fields that we obtained with the local gratings and the receptive fields we obtained with the wavelets. And we just had a meeting prior to this one with Sophie and Max, and we're trying to troubleshoot what is going wrong there. And one thing that we noted was that the overall correlations we get are too low compared to what they expected from their experience. So there's still something in the pipeline that prohibits us to really pick up all of the correlations or the best wavelets, maybe. And yeah, one other thing we noted was that there's still an interesting difference between the different degrees in orientation. So here we have something that's more kind of centered in the visual field, whereas for the 45 degrees we have this weird bias to one corner, and in 90 degrees we have a different pattern, and that's kind of consistent also at the probe D. So yeah, we don't really know what this is, but it's at least consistent between the probes.
Lucas?
Yeah, so this was
Y-
kind of our experience. Maybe you, Jerome, just kick in.
You have a question from Sarah in the chat, and I'm just going to read it because I know she is sometimes not able to speak. Is it possible that the surround has an effect in the zebra noise that impacts the response compared to the classical mapping?
That's a great point. That's also what we thought might constitute some of the differences here, but maybe Sophie or Max can comment on their experience with that. But I would also expect, because it's kind of, you don't have this spatially confined stimulus, but you show essentially a stimulus on the entire field, you also pick up more of the recurrent and indirect drive to that cell. So it kind of also would be expected to give something else. But yeah, maybe Sophie or Max, you have experience with that, or would you say it's at least the maximum of the correlation should still correspond to what you would expect from this very localized, let's say, feed-forward drive?
Yeah, so to answer Sarah's question, so yeah, there would be probably an effect because it's not like an isolated stimuli. You have like a bunch of stuff that are also happening. So especially if you expect the neurons to be locally connected, for example, you would expect maybe to have some effect on what the like how the preferred wavelet is chosen. But in our hands, it never was so bad that you can't properly figure out what's the resonantopic position of the neurons. So what we think is happening here is that there is probably some issue with the way the correlation was computed, and something we're trying to troubleshoot right now with Marcel and Lucas. And but maybe you're going to go to the point later on. But I think there is like maybe a couple of additional experiments that could be made in order to be sure that everything is working the way we're expected. Because I don't think like those matrices that we're seeing on the screen right now are very to be trusted, because yeah, it doesn't look like you're supposed to have a correlation between the wavelet and the neuron that is around like 0.3 or point something, and the one that you found here is like it's like so little that I don't think it can be trusted too more.
We can see it on this slide. We don't have the color map here, but it's this is in analysis. We come to that in a bit, but it's you essentially see the correlation values are on the range 0.03 to 0.07, and Sophie, in the analysis they obtain something like 0.3, so almost an order of magnitude higher, which is quite kind of substantial. So we were trying to troubleshoot what's going on, but we have some directions, but it's a bit open if we figure out. Maybe Marcel, you can just quickly talk about the alignment and delay analysis.
Lucas, Stefan had a question.
Oh yeah.
Quick question on the method. Was it used before on neuropixels? I did look through the bioarchive, and it seemed to have been on calcium, which of course is going to have a very different correlational structure, pretty much due to the method. Sophie, have you tested this on electrophysiological data? Is there something, a preprint for somebody to look at that?
So we tested it, but like it's not on any preprint.
Okay.
I think maybe Max, you're gonna But you haven't really like you haven't done any retinotopy on it. Yeah, but I have like a couple of plots where I've run the pipeline on NeuroPixel probes, and it works actually quite well. But one of the main so should I go directly with what I would suggest to make sure like everything works, or like it's something you want to talk about later, Lucas?
We can, yeah, I mean, I think it makes sense to talk about it. Let's take this.
So what we were like discussing earlier on is like, would it be possible to have like several repeats of the same zebra stimulus? Because then like the type of plots that we usually make in order to make sure that everything is properly aligned, like both temporally and to the stimulus and everything, is a plot where you basically have a scatter of the repeatability or cross repeat for each unit that you get from like kilosvolt versus the depth, like the position on the probes of the unit, and it's color coded according to like what's the preferred position. So if everything is all right, you should see like nothing. Then you reach the visual cortex, and you start seeing like units that are colored, and the color should be sort of homogeneous because since it's a probe, like all the neurons are going to be like retinotopically similar. And then you're like the probe is gonna reach out of the like the interesting layer of the visual cortex, and you should not see so many like units, like repeatable unit anymore. So it's like a nice.
Sophie, if you want, I can also stop sharing and you quickly show it. I think it makes sense to see it.
I'm not sure I can share my screen actually. No, I can.
Because that was quite impressive to see. Yeah.
Do you see it?
Yes.
Okay. So this is repeatability on the y-axis. This is the position on the probes. And each dot corresponds to a unit. The color corresponds to like, so here the preferred azimuth, here elevation, orientation, and size. So it's a nice debug thing because if everything works all right and if everything is done correctly, then each neuron should be quite repeatable across repeats. So you would expect a bunch of like interesting neurons to have like high repeatability. The color is homogeneous, as I was saying before, because all of these neurons are like probably looking in similar areas. And then you would have a bunch of more noisy units where when you don't reach the visual cortex. So it can be like on the upper part of the upper, the lower part of the probes, basically. So it's a nice debug thing to do. Like that's what I would expect if. But this is only doable if you have like a couple of repeats at least. So that's what I was suggesting to.
No, that makes sense. I must say we know that the physiology is okay because the response to grading is strong, and so also that also means the location is okay. So I think I was hoping that the grading will be the strong control. I also know that we have done a longer zebra on the SLAP2 platform with Michael Shi, and I recollect he showed me very strong repeatable responses on SLAP2, which is completely different. But I'm not sure it's the way the stimulus is displayed that is causing this. So I will lean towards analysis being the problem. I don't know what you think.
So we think like there might be like two things. The first, no, maybe three. Okay. So the first that we were a bit like worried about would be like if you only have one repeat and the mouse is somehow like grooming itself or something. Is it possible that it just like hides part of the screen? And I don't know if you guys have behavior videos or something.
Possible. I mean, we did 6 sessions with Zebra.
Okay.
So we have
one should be fine. Okay. The second,
but it's possible that those 5 minutes are unfortunate.
Who knows? Then so it could be a time alignment problem. So what we discussed, it seems that it's carefully done. So for each frame, you have like one timestamp, and you align this timestamp to the one you get from the probes. So that's the way we do it. So it should be fine, but it's always like a hard thing to I mean, that was like the complicated part, like to have like a proper and very precise like time alignment. So that's why a correlation across repeats can be useful, because you would see very well if you have the correct time alignment if you see a high correlation across repeats. So that's the reason why we were thinking like, oh, if you guys could have like at least 2 or 3 repeats for like one, even another experiment where you don't even need to show like anything else, just this. So it's like a 15-minute experiment. So then you can make sure the pipeline for everything like works, that you get proper retinotopy, and then afterwards I would try to apply the same analysis to the data set you guys currently have. So you have like the...
And to add, sorry, and to add one quick thing to what Sophie is saying is that even not for the purpose of debugging, even for the final pipeline, it's probably useful to have 2 repeats when you run the analysis.
Okay, I mean, we did explore Zebra to allow us to map responses in because I understood last time we met that 5 minutes was enough to map the properties, and so now if you want 1 repeat, that means 10 minutes, right? Is that right?
Yeah. Or? So either that or a slightly shorter display of zebra noise with 2 repeats.
But would that be sufficient to map the properties, or?
I think if you're sure that everything is correctly, like here it's mostly for like debugging thing, but then it's also to account for like the variability that you can have, because the you know, like as you know, the like visual related activity, it's only like 20% of like the variance explained of the visual neurons. So it means like if your mouse is doing like something very specific during this time, like if it's running versus if it's like idle, or if it starts sleeping or anything, you can have like a lot of variability in what the neurons are doing. So that's the reason.
Our receptive field block on our pipeline has been 7 minutes long for years now.
Okay.
And we've used it to map receptive field really reliably. In this particular case, it was 5 minutes long, and it was sufficient to do pretty good receptive field. So I hear you. I think you're right, but the repeat is better. But I still feel like it's possible. I think it's possible that there is a delay issue, that we're not locking on the response somehow, and so that we have to debug that. I think this is one of your concern, right?
Yeah, but we looked at the delay with Lucas, and it seems that, I mean, we couldn't find because like they found like a variable delay for each unit, which seems very unlikely. So for us, it's probably just that whatever is like the alignment is probably wrong somehow, or...
Okay, it's possible.
Maybe we can just show the plot.
Yeah.
Do you wanna share, Marcel, or should I share?
Yeah, I'm going to. Okay, so you can see like a cutout of the experiment for probe C and probe D, and the stimuli we analyzed are in this orange pad and this blue pad, so the zebra stimulus and the Gabor stimulus. And here it looks like when we're looking into the changes of the stimuli, it looks like that the alignment and time is all right, I would say. So for example, here when the tribute stimulus starts, we see like on both probes like this jump in activity, and also we see this cut in activity when we are jumping to the zebra stimulus. I guess so. From this plot, I would say the time alignment is okay. What we also did is to have a look whether introducing a delay benefits the analysis of the superstimulus. And this is what you can see on this plot. And I also think we can drive one conclusion from that. So what you can see is we pick the maximum correlation we find for different orientations of wavelets for different values of delays. And we do this for 2 sessions here, and we can see that that the units don't share a common preferred delay, which is very surprising. I would conclude from that that two things there are 2 things. So maybe one main thing we have to change in the analysis mainly is that we have tried to match the analysis of these Gabor filters to the analysis of the wavelet stimulus or the zebra stimulus, and we restricted the wavelet or the pipeline of the wavelet analysis from Sophie and Max quite a bit to do so. So for example, what we're doing, we decompose the zebra stimulus only with wavelets that are comparable to the Gabor filters. And I guess this is something you should do when we're looking back to this. So I still have the hope when we maybe also optimize like filter size, so spatial extent and frequency of these wavelets we're using to decompose the zebra stimulus, that we can have better results than what you're seeing here. Because right now, like for any of us, this doesn't make a lot of sense. Yeah.
Yeah, this is suspicious of timing alignment, because you should get a peak here.
Yeah, exactly. This is what we're expecting.
Alex?
Yeah, so I'm a little lost. I forget the details of how we generate the stimulus, so I'm a little lost on the analysis. So it sounds like, please correct me if I'm wrong, we're actually saving the stimulus frame by frame. Is that correct? And then we use that to compute the wavelet responses. Is that correct? Because I'm wondering if there's something even worse going on. So for example, if the movie that is saved is not actually what has been shown. Because the stimulus generation, the way that we ended up doing is different than what Sophie did. And so it sounds to me that maybe it's worse than alignment. Maybe it's a totally different stimulus, for example. That's what I'm worried about.
So the stimulus we are analyzing for this wavelet decomposition is the video saved in the repository.
Yeah. This should be
so
the
how
one.
do we know? How do we know? Have we checked that is actually correct? That is really the one that it shows. We know that for certain.
It should be, Alex. I can do some sanity check even with feedback. So the way the frame time is tracked is Bonsai loads the movie at the onset of session, and we're using a little object in Bonsai workflow that plays the movie back. And every time a frame is displayed on the screen, the code takes the frame number of the movie and the frame number of the bound vision session since the start of the session, and it logs that. So I know the frame number, but I know those 2 frames number, the frame of the number of the movie and the frame number of the bound vision session. And then in addition, we also have a separate computer that is tracking the time of each frame that bound vision is displaying on the screen at 100 kilohertz. And so this is what's used for all of the overstimuli through the session. So when we package, we take the absolute time from the start of the session of the bound vision frame, and then we use that time into the steam table based on the logging. It's possible something is off. Time alignment is always there's always edge cases that we encounter, and we're constantly making sure that this is correct, which is why I was paranoid about having gradings there and doing this, because I think this is really important. Based on what you're showing, it seems like we need to double check, make sure that make sure that there's nothing unexpected going on, because that peak is troublesome. Is concerning. That absence of peak.
Yeah, exactly. Maybe one thing I can say to this is that at least the number of frames mentioned in the data and also in the video match. So I guess it's the same information there.
The number of frames and the number of frames in the movie is what you meant, or?
Yeah, the number of like entries in the data, so the time steps of the on and offsets of a frame of the movie and then the movie itself. This is the same.
Yeah. It could also be in your code, how you read the data. I'm not sure. Maybe not.
No, I'm pretty sure that so I also tested how to read in the video and so on. I think that that works. I think one thing we definitely have to try out is to get rid of the restrictions we put onto the pipeline of Max and all of the original work. Because I think in the end, yeah, you don't need that to make the analysis comparable. So the analysis of the zebra video and the analysis of the Gabo filters. But we didn't. We thought differently when we started the analysis.
Okay. Sarah is asking, maybe we could use a photodiode to check luminance. I mean, we have a photodiode in the corner of the screen that's actually used for alignment. It will be a bit complicated. Yeah. I mean, it depends how much time we want to invest into it, because we have Zebra is auxiliary to our goals, I think. And we have the whole, because I think so far, some of the analysis that Ali with Fazane have shown support that they are responses, and they are all super lock to what we are showing on the screen. So all of the stimulus from the beginning until Zebra are like on time. And we know there are oddball responses, so I feel like the rest of the design is okay. There's this part in the Zebra and the Zebra and also like there's some weak opto tagging response. They're not great, but they're also aligned. And so it's possible that something particular with movies, but yeah. I think
so maybe to move forward. So there was just one result where we had the feeling, okay, it might not be entire like just noise. For example, let's say there's a problem with the assignment either during analysis or data collection. But there's one thing that maybe Marcel can go to that slide where we have different correlation values for the different orientations, which kind of matches also a bit what we saw in terms of the spatial layout. This is never mind this plot. So essentially, it's centering the response at this optimized delay for each unit. So this is essentially the best delay per unit. It's just that if you look across all units, it's not the same. So we don't see it in the mean over all units, but per unit, you can pick up the best one. You could also do that with noise, of course. So this might also just be optimizing noise. But what you see is that the level of correlation is just different between these orientations, which is a bit puzzling to us. But yeah, so I would say we have with Sophie and Max, we just identified few things that we could do on the analysis side to just try to see if we get bigger correlation and a proper delay there, which is ones using different spatial frequency of the wavelets, not only the ones that we use for the Gabors. Sorry, for the gratings. So playing with that parameter, we want to change the response window. So right now we average spiking responses essentially per frame, just in a time-shifted manner. And that's something we want to see if we extend that window and average over a longer period. Would that increase the correlation? Yeah, that's essentially things that we want to check. That's something we can get, soon and distribute that in the discussion, and then we can see if this changes, the results, and we get something sensible there. But from now, it's hard to conclude if there's something in the analysis pipeline. I would also expect that the temporal alignment is kind of fine, and it should be something in the analysis. That's my guess.
Just a quick, have you tried, so we saved the movie frame by frame. Have you tried to look at it? Have you tried to put it back together and look at the movie if it makes any sense or it jumps or?
Yeah, I looked at it frame by frame. So I loaded frame by frame, and you see like the movement of the movie, and it corresponds to when you play the original one.
Yeah.
Mm-hmm.
I mean, I can spend time today and tomorrow just reviewing the alignment process to make sure that nothing finicky sneaked in. Also, is there any? I mean, is Max and Sophie? Do you feel like have you reviewed the code from Marcel and Lucas? Do you feel like it's good or?
We saw the parameters that you're using and the overall structure, which I think is fine. Maybe for like something, something I forgot, like, so I know you want to match the spatial frequency with the one that is displayed on the Gabor experiment, but I was thinking like if you use at least like two spatial frequency, like one maybe a bit lower, just to see if it makes any difference. I don't think this is the problem, but it would be nice to have it. And otherwise, the only thing I could that would really help to debug like what's going on would really to have like two repeats and try to see like how they are to like if they look like if they look similar or not.
Yeah, you have no other way to by looking at the responses know if the cells are responding to it?
But
not really.
I don't know how you would do this, because then you would need to find like because you would need a prior on what the cell prefers. So you need like a proper resinotopic mapping, and then you would need to like find the frames that show this and see if the response of the neurons.
But we have that. We have Gabors.
Yeah, okay, that's true. Yeah, so maybe that's something you can use actually if you don't want to do another repeat. So you just figure a specific neurons for which you know the position and orientation. You try to find frames for which this is, the neuron should respond, and just see how it looks like. And then if you align the two, you try to see if you see a peak in the neuronal activity close from like when the frame is happening on the screen, and you try to see if there is like an alignment problem, something like this. That could be something you could try, yeah. You see
What
what
do
I
you
mean?
think, Marcel and Lucas?
I mean, I was just wondering if you would really expect that response to occur, because it's really, I mean, as Sarah pointed out, it's really different stimulus in a sense. So it would be hard to figure out if, you know, let's say if there's no response, or response shifted in time, that could be due to recurrent interactions, you know, leading to an earlier response, or inhibition leading to suppression of a response, or so that otherwise you would expect from the local grading.
I don't
That's
think you
a
would.
bit my worry, that might be hard to conclude from one stimulus to the other. But yeah.
I mean, ideally, as I said, I think the best way would be to have a couple of repeats. If you don't, I think that, I mean, I'm not expecting to see like no answer because of like surround suppression or anything like this, because we compare the results, like the retinotopic and position and the orientation of the zebra noise with like drifting gratings, like full screen and sparse noise, and it gives similar results. So I don't see why like localized Gabor would be so different.
Can you unpack similar results?
What do you mean?
How different they are?
Oof. We need to load the paper because I don't know them by heart. But basically, it's like the same orientation roughly. So then it depends, like if you have the same number of orientation, of course. Like if you display like 18 different angles on the drifting gratings and you only analyze them with 8, you're not gonna have like the exact same result, but you're gonna have like the same, roughly the same angle. And retinotopic position were actually like pretty much smaller and even less noisy with the zebra. So I wouldn't expect to see something like dramatically different. So I think that could work. I think what you could do is actually to just, like pick according to your localized Gabor the preferred position of your neurons. Try to find like which frames should trigger a firing, and just see if you see like spiking like happening like after like where I would try to just like see if there is like any time shift that this seems weird.
Okay.
But ideally, like a couple of repeats, to be honest. It just makes everything easier to debug. But
Alex?
Well, can I directly ask a question to that?
Yeah, please.
Okay, I do not really see the difference to what we've done, to be honest. So because we tried to do or to build or to construct the filters we are using for the decomposition of the stimulus to be as much as similar as possible as the filters of the Gabor stimulus. So basically what I would expect to see then is exactly what you suggested. That basically you get then the correlation of the neurons firing to whether the frame and the position of the or the cutout of the video corresponds to the filter or not. So I think we've done that.
Yes, but if there is a alignment related, like time alignment related problems, it's going to be very hard to spot, no?
I think Sophie was pointing at, selecting the specific frame where you would expect the max response. I think that's, yeah.
I mean, yeah, of course, we can also pick out single frames and have a look on the activity, yeah, and not the mean activity.
So one thing you could do is the opposite, right? So you could look at when the neuron, let's say, is 2 standard deviations above the median or something in terms of firing, pick those frames and average those and see if anything comes out. Now, if there's an alignment difference, or what I'm really concerned about, if it's an entirely different stimulus that was shown, then that would still be noise. But if you're concerned about the particular parameters or wavelength or whatever you choose, if you do it the other way around and you pick when the neuron is most active and average those frames, there should be some kind of structure in the frame.
Yeah,
The
so
image.
this is the spike triggered average you suggest. We also tried out that. The problem here is when you, like on an average, you have a sparse firing. So you don't have many frames to average. And then like the average stimulus is extremely noisy, and there are very not a few amount of instances where you can see a nice Gabor-like response.
So to jump on this, the spike triggered average could give you like interesting results, but only for like a small subset of neurons, because it would only work if you have like linear cells, like simple cells. But if you have like complex one that have like several phase, like a broader phase tuning curve, then you wouldn't be able to see anything with the spike triggered average, because basically you would get because since it's a zebra, so the neurons would both respond for like when like a specific black and white pattern, and also the other way around, so the same pattern but with inverted colors. So it would like sort of average everything out, and you wouldn't see anything for those complex cells. But for the subset of cells that are simple, you should be able to see something. But it's just going to be like a much smaller amount of cells. So I don't know if it's.
I mean, we have some beautiful examples, but they are very few.
Okay.
Okay.
Yeah.
So there are some
Jerome,
unknowns.
last question real quick. So could contrast play a role? What contrast level did we end up choosing, and how does it relate to the one that Sophie chose?
Oh man, I thought it was full contrast. No. I don't think I play with contrast here. I mean, I'm gonna check that I'm gonna double check that the movie that is on the repo is the right one. I think I was careful there, but mistake happened. I can check the timing code to make sure there's no bug here. Although I would expect your delay plot should show something, because if it's
weird. So yeah, I mean, we will also double down on that side.
And have you sorry,
anything.
Lucas, have you done this on more than one mouse?
We have a second session. We just had 2 sessions from the same mouse.
2 probes from the same mouse.
And 2 sessions also. 2 probes.
Oh, I see.
Sessions. Yeah.
In both cases, you see strong Gabors.
Yeah.
Oh, that's the Gabors we just checked now to see if these troubles we have here appear also in the other session. And these plots, for example, they look the same for the other session. We didn't check the Gabors in the sense of this slide we showed before.
See.
But I'd expect we find something. So we will check and post the plot.
I think we did the comparison of Gabors in the very beginning, and they basically gave the same results. Also, we find a similar number of significant responses, for example, in the Gabor stimulus for different sessions.
Yeah. I don't know if you have any time, Sophie and Max, but the data is public, so if you want to take a look, also will be wonderful.
Yeah, definitely.
Yeah.
Yeah.
Okay. I have one question about the like the schedule in general.
Yeah.
So I understand that you want to have this analysis as quickly as possible. And you said, okay, next week you want to go to production, right?
Well, what was the ideal case? Because we have some mice that are ready to go, and now we have some uncertainty about zebra, so we have to sort of make decision in that context. I can make modification to the session, but everything else so far is good. Like the response to oddball, the response to Gabors. Severin has done analysis of the targets where the probe went through using histological reconstruction, and there's just one probe that needs to be adjusted to target the ACA, or actually the MO, more directly. But we essentially went where we wanted to go. So in terms of targeting, we all 90% good. So it's really about, the only uncertainty is about zebra, but all the rest is good. And so I don't know if we have the time to make an iteration here, or we just keep the stimulus as is and just make sure that there's no issue with the moving being shown, so that later on you guys could figure it out. Because I don't think well, for sure this is not calling for us to remove Gabors. Do we agree? So we should keep the Gabors. Then I would advocate for just keeping zebra in and making sure that nothing wrong is with the zebra as we display. It's the right movie. We keep track of time properly, and then move on. And then as we collect the data, you can analyze it deeper, and then leverage it. I don't think I wouldn't advocate to making big change here. What do you think?
So originally I wanted to ask whether we should, of course, as to get this tests done on the analysis side as fast as possible, but there is then a hard deadline would be next end of this week, for example, when you want to start next week.
Well, it's just that I think what we want to avoid. So there's always unknowns. Like, neuroscience is complex. So there's always unknowns when we're coming in and experiments. And so what I want to avoid is data integrity issues. So if you have concern about timing for zebra stimuli, I would spend time the rest of the week trying to figure it out, like make sure that there's no problem on our hand, so that when we collect the data, you can still analyze it. Like later on, you're smart guys, you're going to figure it out. Like, where is the problem in the analysis? But we don't want to have problems in the data collection, because that's impossible to correct. And so that's the goal of the pilot, right? Is to make sure that nothing is wrong there. It look like you're pointing at potentially a timing problem, which is in our hand to fix. And so I would, So based on what your feedback is here, I would advocate, I would spend time trying to make sure that the timing is correct. So that's going to be work on the rig and checking everything is correct. But but it doesn't seem like Zebra so far has given us the nirvana we hoped for. But maybe we can get there.
Yeah.
I think that makes sense, Jerome. The only question is, as Sophie pointed out, it might make sense to change the zebra in the sense that we introduce a repeated version. I don't know if 3 minutes is too little to still be useful, but if one could have 2 times 3 minutes or so, just for future analysis, so that we have essentially this repeated.
Yeah, we could do that. But is 3 minutes enough?
I would. I don't know. I think for some units you're gonna have like not very reliable because some neurons are sparse firing. So I would expect some like visual responsive neuron not to respond just because they are like they're sparse, and that might be missed in the stimulus. Ideally, it would be 2 times 5 minutes.
I see. Yeah, it's just the longer
Jerome?
the session. Yeah. Yeah, Alex?
Jerome, what if we drop Trippy and just repeat Zebra instead?
That's a good idea. What everybody thinks about it?
The rationale to include Trippy was to have kind of a physiological fingerprint, right? So that we can map to the microns.
Yeah.
Yeah, that's right. That's right.
I mean, one thing, if you guys have time, you could, theoretically speaking, run your analysis on the Trippy also, right? Because you have the same frames. It should be pretty much the same analysis, at least theoretically. So if something went wrong with the Zebra stimulus and it works with the Trippy stimulus, maybe you get something out of that. But if both are misaligned or something is wrong with these video frames, then we might be wasting 10 minutes. And at least if we do 2 times Zebra, we might be able to find something out about what might be going off. Or at least also it might be interesting to get that additional measure of how repeatably the repeatability of the responses of those neurons.
Well, I mean, we got far more engagement from the Zebra community than the Trippy community. And so I would advocate for giving more time there. Is there any concern with us doing that, or? Because I think Alex is right. We might be trying to do too many things.
I mean, if we
one question would be what we would want the Zebra for, as kind of just to open that question as well. Because if we stick to the receptive field gratings, we have the receptive fields, we have the orientation tuning from the controls in principle. So
we don't have spatial frequencies, right? We don't have spatial frequency tuning.
Yeah, we got spatial frequency tuning. There's this more elaborate analysis of kind of the speed tuning as well that one could pull out of Zebra.
Now, how about this? If we do half the sessions 2 times Zebra and half the sessions Trippy and Zebra, what I could foresee is if we work everything out, we might be able, because Trippy and Zebra are such similar stimuli, we might be able to find out how neurons convert. And then we could say, look, what we get out of Zebra relates to this response in Trippy, and therefore we can relate it to the microns data. So we might be able to get both out of the best out of both worlds, but maybe we should split the sessions where half of the time we do Trippy and Zebra and half of the time we do double Zebra.
That's getting complicated, Alex. We already have two number of cohorts. I mean, the challenge with Trippy is we're not even sure we have the exact same movie.
Yeah, I'm worried about that too.
Whereas I kind of like doing double Trippy because double Zebra because it strengthens the statistics. And so if we get if eventually we figure it out, when we get all the tuning properties, that's going to be helpful because we actually did the work together to fine tune Zebra to give us both E and I responses. So I feel like it's a better investment.
And I think one other thing, and this is a point that Alex had made before, is that you also get the repeatability measure when you have two of it. So even how responsive is it to visual stimuli if you have 2 repeats, which can be interesting on its own.
Yeah, especially in the context of predictive processing. It might be a really interesting variable, yeah.
Okay.
That sounds like a plan.
So I'm gonna write this up in the forum, and then if anybody object, we'll see there. I mean, can we continue to engage on this analysis offline? Because I think it will be, yeah, we might figure it out eventually. But I would, so I'm going to spend the rest of the week potentially doubling up Zebra, and then we'll use that for production. No concern there? Or?
Yeah, I think if you take an iPhone movie of what is shown on the screen and then we replay and it's the same, then I think we can reconstruct any alignment problem. I think the only issue would be if what we believe has been shown on the screen, even though we get the time not right, was something else entirely. But if there's any lag or shift, then we should be able to reconstruct it, right? So the only thing we have to make sure, because this can happen sometimes if you pseudo-randomize data or you have lookup tables, that you get a shift in the trial count. And so I've seen that before. So as long as we know it is showing what we think it shows, I think we should be able to in the future reconstruct the responses.
Yeah. Yeah. Okay. Any other comments? Sorry, we went over the hour. But I'm yeah, incredibly thankful for all the work you did, Lucas and Marcel, and also thanks Max and Sophie for jumping in and sharing your wisdom there. It's invaluable. Let's continue in the forum, and then next week we'll have John Meng present his theories. Thanks, everyone.
Okay, see you. Bye.
Thank you. Bye.
Thank you.